Penguins. So today we are going to work on natural selection. So if you're an AP Bio and you're trying to, get ready for the exam that is in what, 20 days from now, you're in the right place because we're going to go over Unit 7, which is one of the larger units that is on the AP Bio exam. And so like when you think about like the levels, like the different units we have, Unit 3 and Unit 7 are the ones that have the most. So we definitely need to make sure that we have Unit 7 down. So you may be wondering, why did I start with AP Bio Penguins? Because of the fact that you sitting in this session as an AP student, you are now ready. You're going to be prepared for the exam. So I like to say that AP Bio Penguins address success because no matter what happens on the exam, you're prepared, you're ready, and you're going to rock this exam. So in case you don't know, I do have daily review that I do on my Instagram. At this point in the game, where I think I just did 8.2 on Friday, so I'll be on 8.3 on Monday. But I do have all those past reviews all saved on my website, so you can easily access those. I also have a 374-page review guide. Again, at this point, you don't want to deal with the whole 374 pages. What you probably want to look more for is spot checking. So any topics that you have trouble with, you would go to those specific units, as well as at the end of each unit, I've got multiple choice and free response practice. And that's really where you want to focus right now, is doing those practice multiple choice questions and doing those practice free response questions. I have a record FRQ Friday on every single FRQ going from 2013 to 2023 on my website. So after you do any of those FRQs and you want to kind of get a little bit of feedback, see how another student may have written it, I've gone through every one of those FRQs with you. I also have 120 review games on my website. So if you go to the page, there'll be just a little code. You then go to quizzes.com and you type that code in, and you'll be able to access that game to review, as well as I've got review PowerPoints plus, of course, all these live streams. So today's play, we're going to work on natural selection. We'll talk about Hardy-Weinberg, and we'll go through phylogeny and then like evidence of evolution. Then we'll do some practice questions, and then I'll give you a chance that you can do some Q&A. Now you are able to type in the chat, so if you have a question as we're working through this, you're welcome to type it in there. I do notice that there is a slight lag between when I say it and when you type it. And like when I see it. So if I don't get a chance to answer your question, I'll probably get it before the end of the slide, but I have noticed that some of y'all like to answer each other, which is very helpful. So if you see a question that you know the answer to, and I'm in the middle of talking, you're welcome to answer each other's questions too. So let's get started. So first thing, what is natural selection? So natural selection was created or developed by Charles Darwin. He was on, the SS Beagle. He went over South America, and he started noticing all these different similarities between the organisms that were in South America, as well in the Galapagos Islands, as well as over in like kind of where he was from over in Europe. And so he came up with this idea of that we have descent with modification. So each of our descendants are going to see slight modifications due to different traits that may have been more favorable. Okay, so the organisms with the more favorable trait, they're going to be more likely to survive. And because of the fact that they're more likely to survive, they're going to be more likely to then have more offspring, and those offspring will also be more likely to survive. And so we'll see that our gene pool, we'll see that those alleles in that population are going to increase for the favorable alleles, and they'll decrease for the unfavorable alleles. Okay, so we have a couple different examples that we've seen of this, like the peppered moths. So if we were to assume that these little beetles are moths, which of course I know they're not, but let's assume that they were, what we're looking at is that the individuals that are able to kind of like in this environment, they can blend in. So that was what kind of happened with the peppered moths, is that the pollution from like when we went through the Industrial Revolution, the pollution then caused like the moss that cover the trees to die. And so the trees were a darker color. And so the beetles in this case, or the moths in my example, were more likely to kind of blend in with their environment. And so if they can blend in their environment, they can camouflage, they're less likely to be consumed. And so they're more likely, of course, to survive and pass on those traits. And so we can kind of see that over time, those individuals that were not consumed, of course, can reproduce, and we see a change in the allele frequency. So if we were to say that the dark allele is our capital A and the light allele is our lowercase a, you see the frequency has increased because this is a more favorable trait. We also see this antibiotic resistance. So the bacteria that have the resistance to antibiotic, you take the antibiotic, you kill the ones that are not resistant. Now we see that there is a higher chance of the probability of, or a higher chance of the population of bacteria. Now, of course, having this resistance, so we see an increase of that antibiotic resistance. So oftentimes you'll see that they kind of will alternate between different antibiotics, as well as they may kind of hold off and let your own immune system kind of fight the infection on its own. But when we do think about natural selection, we do want to be careful of Lamarckian statements. So Lamarckian tried real hard, but he was wrong. So he had this theory of use and disuse. So, and I gave this example to my students earlier this week because they were having trouble with one of the questions that they saw in the practice exam. And so the student was like, I don't understand. Like, if this thing happened in my lifetime, then of course I can pass it on to my kids. I'm like, if I cut off your ear, is your kid going to be born without an ear? And they're like, well, no. I said, well, that's the same thought process of what was happening with Lamarck. Okay. His thought was that if something happened to you during lifespan, that it would then get passed on to your offspring. The only way that you can pass something to your offspring is in your gametes. There has to be a genetic component, so some type of mutation, some type of change in your DNA, and then that has to be in the DNA of your gametes that goes on to that generation. You having purple hair is not going to cause your child to have purple hair. So you do have to kind of watch out for those different Lamarckian statements as answer choices in these evolution questions, as well as you need to make sure that you don't say these Lamarckian statements when you are like kind of answering a free response question about it. Artificial selection is that organisms that have a specific trait that we want, they're going to be, we're going to kind of breed them and try to have more of them. And I could never figure out how to undo my face. So I'm sorry. I think I am going to cover up this font. I do have the whole PowerPoint posted on my website. So I'm sorry that my camera covers it up. But so we have different traits that we, of course, bred because like they were something that we liked about them. Okay. So for example, dog breeds. There were certain dogs that had certain traits and we thought those were great traits to have. So we bred those dogs to have that trait. We kept having more of those. We made sure that we bred the dogs so that they would get certain traits. Like there were some that were for hunting, and then there were other ones that were supposed to kind of draw out other individuals. I think like the weasel dog or the wiener dog, its job was to kind of burrow and get the different kind of rodents that were underground to come out. We also see this with wild mustard. So the wild mustard, they took different parts of it. So like the apical stem, the leaves of it. And that's where we got cauliflower and broccoli and cabbage and kale and calabre. And so like humans are actually impacting this. And a lot of students think that antibiotic resistance is technically artificial selection, but it's not. Because although we are taking in antibiotics, it's part of the environment. It's part of this the bacteria's environment. So it is technically natural selection. So here we can kind of see originally we had the wolf, and then of course through artificial selection, we've selected for certain traits. So there we go. So we also see that we have three different types of kind of selection that can take place. So we have disruptive selection. Disruptive selection has to do with if this was like where the population was. We had mostly the intermediates. What's going to happen is it's going to disturb the middle. It's going to make it so that the two on the outside kind of become the prevalent one. They make them more extreme. And so like let's say, for example, we have a small beak, a medium beak, and a large beak bird. And then a drought happens. And so the seeds that the medium-beaked bird used to consume is no longer around. And so all we have are the berries that the small-beaked bird can eat, and then these really hard shells that the large-beaked birds can eat. And so because of the lack of that food, the medium-beaked bird is no longer going to be able to find food, okay? It's not going to be able to compete with the small-beaked bird to get the fruit, and it's not going to be able to compete with the large-beaked bird because it can't break open those shells and get the food. And so we're going to see a decrease in the medium-beaked bird and an increase in the small and the large-beaked birds. Okay, that's disruptive selection, so we select for the two extremes, and it's against that intermediate. Stabilizing selection is where we're going to select for the intermediate. We're selecting for whatever's in the middle of this, okay? So the intermediate phenotype. So example we could use for this is birth weights. I don't know about y'all, but I was about 6 pounds and 15 ounces when I was born, and then my kids were somewhere in the 7-pound range. And you were probably somewhere in that 6 to 8 pound range. The reasoning why is because back in the day, now we're thinking evolution. Back in the day, if the baby was too small, it's probably not going to survive. And if the baby was too large, it also was going to have trouble coming into this earth and probably wouldn't survive. Because again, this is before modern medicine. And so we saw that kind of stabilize to that kind of 6 to 8 pound range in the middle. And then, of course, directional selection. With directional selection, it's moving towards a phenotype. There's something about this phenotype over here that is more favorable, right? And so if we were to think about, I don't know, horses. Back in the day, horses were kind of small. And then we started using them for farming. And so we saw that there was an increase in the kind of the size of the horses and the robustness of the horses. Now, again, be careful of the marketing statement there. It's not because we used them a lot that they became larger. It's because the ones who were able to carry the farming and were able to be robust, they didn't die when we used them. And so we were seeing kind of that changing in the directional selection of those horses. Okay, so Hardy-Weinberg. Hardy-Weinberg is a theory, okay, and it's based on these 5 different characteristics. And if these 5 conditions are true, then the population is not going to evolve, okay? So Paul Anderson from Bozeman Bio, he did a TED Ed video. It's called The 5 Fingers of Evolution. And it's a really great video. It's maybe like 5 minutes long. So if you are struggling on Hardy-Weinberg, look up his video. Again, it's The 5 Fingers of Evolution. It will look like that. This is like the kind of the cover art looks like. And it helps you to really get down these 5 because he's using the fingers of your hand to come up with these different things. Okay. So number 1, we need to have an extremely large population size. Having an extremely large population size will account for any type of genetic drift. Any random thing that happens in your environment, like a natural disaster or whatever, isn't going to drastically decrease your population to the point that we lose our allele frequencies because genetic drift is just random things that changes the allele frequency. Okay. We need there to be random mating. There cannot be selection for or against a certain trait. It needs to be random. That way we're able to continue to pull from that same gene pool. We want there to be no mutations. If we have a mutation, that's immediately going to change the allele frequency of my alleles, okay? And so we're going to, of course, see a change. And then we want there to be no gene flow. No individuals can come in or out of my population. So no immigration and no immigration. And no natural selection. No traits can be more favorable or less favorable than another trait. And so if these 5 conditions are true, then it's thought that evolution is not going to occur. But if one of these happens, then evolution is occurring. So how do we know that evolution occurred? We're looking for a change in allele frequencies, okay? So as I said, genetic drift, there are 2 different types. We have our founder's effect. This has to do with, and I think it's actually founder effect. I like to put a apostrophe S on it. I don't know why. It just sounds better when it's like founder's effect instead of founder effect, whatever. Anyways, so the founder's effect, this is where we have a small population that gets isolated from a larger group. Okay. And so we have this big group of individuals and then this small little subset just happens to go over here. Okay. And they make their own new population. Well, the small population that went over there is not going to be representative of the original allele frequency. And so we're immediately seeing that major change in allele frequencies in that new population, okay? We also have the bottleneck effect. Bottleneck effect is when we have this large population, right? And then some natural disaster, something happens that drastically decreases my population size. So when we had this natural thing happen that decreased my population size, it wasn't selecting for or against traits. It was just random which individuals survived and which ones died. And so again, our new population isn't going to be representative of the original populations. So both of these are going to change my allele frequency. And so any type of change in the allele frequency or genotypic frequency means that evolution is occurring. So on the AP exam, they may ask you, is this population evolving? And so you would have to think about, is the allele frequency before and after the same? If the allele frequency is at all different, then that means population did evolve. Okay, so in our formula that we'll use for this, there are 5 different variables, okay? There's P, there's Q. P is going to be your dominant allele, and Q is your recessive allele. Now, on the actual formula sheet, I think it says allele 1 and allele 2, but traditionally when we talk about this, we talk about P being our dominant allele and Q being our recessive allele. If the question you have is an incomplete dominant or a co-dominant question, then you could just arbitrarily pick P and Q because they're not going to ask you what is P. They're going to ask you what is the frequency of the red allele or whatever. Okay, P squared is going to be the frequency of homozygous dominant. Okay, so that would be having 2 dominant alleles. And so if I have P times P, that's just P squared. That helps you to remember what P squared is. 2PQ is going to be the frequency of my heterozygous. And remembering that heterozygous have one dominant and one recessive allele, so P and Q. And of course, if you were to look at a Punnett square of a monohybrid cross, you know that this square and this square are both going to be your heterozygous individuals. And based on the fact that I don't think it's mirroring, you know which diagonal I'm talking about. And then Q squared is going to be the frequency of my homozygous recessive. Okay, so again, Q times Q is Q squared. So that's going to be your homozygous recessive, so having two of them. Sorry, I got distracted by the fact that I'm like a moment behind because I'm watching the chat just in case I got distracted. Sorry. Okay, so in the formula we have on the formula sheet, we have P plus Q equals 1, and then P squared plus 2PQ plus Q squared equals 1. Okay, so these are the two formulas we need. Now, whenever you're solving this, you always want to start with Q squared. So they may tell you the frequency of the dominant phenotype. Well, dominant phenotype is going to be the P squared and the 2PQ. So you can't just plug that part into this. You have to say, well, 1 minus that to get it into Q squared. But once you have Q squared, you can go from Q squared to Q by taking the square root. And then you can say 1 minus Q, which will give you your P. Once you have P, you can find P squared. And then once you have, of course, P and Q, you can find 2PQ. And so I usually use this little chart, and I tell my students that no matter what they're asking, you just go ahead and make this chart and fill out this chart so that you have all of the information they may be asking, okay? Now, there's a second formula. Now, I have 100% just made up this formula. There's probably an official one somewhere. I've never found it. I call this the counting alleles method. I don't know what anyone else calls it, but that's what I call it. And in this case, you're given the number of individuals that are homozygous dominant. You're given the number of individuals that are heterozygous. You're given the number of individuals that are homozygous recessive. You know how many of the 3 different genotypes. And so in this case, you have to count the alleles to figure out your P and Q. So to do this, you're going to take 2 times however many of them are homozygous dominant. Because if you're homozygous dominant, you have 2 of those dominant alleles. So 2 times that will tell you how many dominant alleles are in the homozygous dominant individuals. And then you're going to add the number that are heterozygous, because each heterozygous individual has 1 dominant allele. And so that will allow you to count how many dominant alleles you have, and you'll divide it by the number of individuals times 2. Why did we multiply by 2? Because each of our individuals is diploid. They have 2 sets of their chromosomes, right? And so they'll have 2 of every allele, or of whatever alleles, okay? So we'll take that and divide by the total, and that gives us it, okay? Same thing with our Q. We would do 2 times the number that are homozygous recessive, because of the fact that they have 2 recessive alleles. And then we're going to add in the heterozygous because they have 1 recessive allele. That tells us the number that are recessive alleles over the total population times 2, which is the total alleles we have, okay? And then P squared will just be super simple. How many of them are homozygous dominant over the total? Heterozygous, how many of them are heterozygous over the total? Q squared is how many of them are homozygous recessive over the total. So that's super easy. A couple years ago, there was a question that they had where they were doing a chi-square on this. And so if you were to have to chi-square it, you're going to have to compare what you get from the Hardy-Weinberg equilibrium and how you would get it from the counting alleles. And that's going to determine whether it actually is in Hardy-Weinberg, okay? So did the population evolve? This is going to say, okay, if the allele frequency or the genotypic frequency changes, then the populations evolve, which I've already mentioned. Let's do a quick practice question on the formula just to make sure we understand how to do it. So there's a garden, 200 garden at your school always has red, pink, and white snapdragons. Those are my favorite flower, y'all. Have you ever seen a snapdragon? They're amazing. If you've never seen a snapdragon, they have this little mouth on them, and you just like squeeze them, and they're super cute. Anyways, there's 200 red flowers, 300 pink flowers, and 500 white flowers. Determine the allele frequency of the flower allele color. So since I know that there are 200 that are red, I'll say 200 divided by 1,000, and that tells me that my P squared, my homozygous dominant, or my RR, because it's actually incomplete dominance, my red allele squared is going to be 0.2. And then determine the frequency of the pink. This is going to be 2PQ, so 300 divided by 1,000, give me 0.3. My white would be Q squared, so 500 over 1,000 being 0.5, because we had 500 that were white, okay? Now, that's not usually where you get confused. Where you get confused is down here. So when we're trying to find the frequency of the red allele, we'll do 2 times 200, because there were 200 that were red, plus 300 because the pink flowers have 1 red allele. and then we'll divide by the 2,000, giving us 0.35. And then if we were to do this with the white alleles, so 2 times the white individual being 500, plus the 300 being the pink, that gives me 1,300 over 2,000, giving me 0.65. So you're like, okay, did this match what we would have done with Hardy-Weinberg? So if you'd done it with Hardy-Weinberg and said, okay, well, let's start out with Q squared. Q squared being my 0.65. I did this wrong. We're going to go past that because when my quick trying to fix this for the review session, I went ahead of myself. Sorry, y'all. Anyways, so phylogeny, different evidence that we have for evolution. We've got our biochemical as well as our morphological. Okay, so different evidence we can use is that if you use like your DNA or your proteins, that's going to be a better way to determine whether evolution, like kind of evolution is occurring. You can kind of say that these two are more closely related because of the fact that we're looking at the number of allele frequency differences, or we're looking at the number of amino acid differences or our nucleotide differences. And so between DNA and protein, DNA is going to be a better source because of the fact that you can have silent mutations. Mutation could be taking place in an intron. So there's a lot of places where the DNA could change that isn't actually going to change the protein structure. And so between those two, DNA is more reliable. Okay. And then you also have morphological. So you could be looking for similarity in traits. So similarities of traits. So you could be seeing like that they have similar bone structures, or you could be seeing that they have similar embryonic development. So for example, did you know that while you were developing, you actually had a tail until, of course, apoptosis took over and you like lost your tail. That's why you still have a tailbone. It's because our ancestors had tails. And so there could be ancestral traits, things that we got from our ancestors or from different descendants along the way. Okay. Derived traits are the ones that come from our descendants. Ancestral are the ones that come from the ancestor. Okay. You need to be careful of analogous structures. Analogous structures has to do with things that look similar, but they have no common descent. So sugar glider and a flying squirrel. A sugar glider is a marsupial, and a flying squirrel is a mammal. Is a placental, I'm sorry. And so if you know anything about marsupials, you know they're in Australia, you know like the koala, and you know that they go through premature like birth, okay? So they're born, they're itty bitty, teeny tiny, and then they develop inside of a pouch. Versus placentals, they are full birth, okay? Full term. And so they're not related at all. But they look very similar because they both have this little thing underneath them. So that kind of shows you, oh, that's similar. So those would be analogous structures. So just be careful because they have similar adaptations to a similar environment, and that leads them to having these similar structures, but there's no common descent. So someone in the chat says they've never seen that second formula before in any of their reviews, in any of their stuff. I know. So I didn't really like think about this until I was doing an activity with my students a couple of years ago, maybe like 10 years ago, but in which we had to count the alleles, and I discovered that there were slight differences. And so keeping in mind that Hardy-Weinberg is a, like it's an equilibrium, it's based on a theory. So now the counting alleles method is a better method to determining the number of each of the things. But if you don't have enough data to be able to count your alleles, maybe you only know how many of them are recessive, or maybe you only know how many of them are the dominant phenotype. And so because of that, you cannot determine exactly what the other ones are. And so that's why this is just more of an approximation. It's an estimation based on things we've seen versus this is a actual number counting. Okay, so don't be worried. You know, it's a logical kind of equation. That's why I said I made this equation up. You could probably write it any way you wanted to, but it's not on the formula sheet. So I did want to just bring it up to y'all. Okay, so Hardy-Weinberg can be in either FRQs or multiple choice. Like there's no determination where it's going to show up on the exam. We just know it could show up. It's in the standards. It's able to be there. So also biogeography. So individuals that are in a certain area, we could say they may have diverged from one another. They could have gone through that speciation event. They could be related because of living in similar environments. So how can we show this? So we can make a phylogenetic tree. So a lot of students have been asking what's the difference between a tree and a cladogram. They show very similar things. This is a tree. You can kind of see it's like rooted here, and it kind of like branches out, and it looks kind of, if you look sideways, it looks like a tree. And so this was the one from, I think, the 2018 exam, maybe 17, I can't remember. Anyways, doesn't matter. In which we were looking at different bears. So like black bears, European brown bears, the polar bears, the North American brown bears, and the Asian brown bears. And we were kind of seeing how long ago they diverged and all that. Okay. And then we also have a cladogram. So a cladogram is going to kind of look like this little Y-shaped thing. And again, it's just showing you different characteristics. And on this exam, they actually wanted you to tell them kind of where you would have placed these organisms. So if we look at this chart right here, this table, okay, what you're looking for is where is the smallest number. Always start with the smallest number. And so I see that the brown bear and the black bear, when they kind of connect, the square that they connect with is a 1, which shows me that there's one amino acid difference in the list protein between the brown bear and the black bear. So since you see how small this little Y shape right here is, I'm going to put the black bear and the brown bear there. Okay, and then I see that the black and brown bear are 7 and 8 away from the polar bear, but the black and brown bear are 33 and 34 away from the panda. So I know that the polar bear is going to be next because they are most closely to the polar bear than to the panda bear. And if you know anything about panda bears, you know those are actually more closely related to a raccoon than to actual bears. So what they can also say is asking you about where could you have rotated the axis. So our polar bear and our European brown bear, they share this branch point right here. And so you could actually rotate along that branch point. And so we could have had polar bear here and the brown bear down there because of that rotation point. You could also see it here between the Western North American bear and the Asian brown bear. Okay, and so you were able to just switch those points. And so when you're doing the graph down here, I picked black and brown, but you could have actually done brown and black because they share that branch point. That branch point is showing you the common ancestor between those two. And so if I was looking for the common ancestor between all of these brown bears, I can see that it happened about, I don't know, 3.4 hundreds of thousands of years ago. Keep in mind that this axis is going this way. So don't say that that's 4.6 because this is a 3 right here. So it's actually going 1, 2, 3, 4. So watch your axis when you're reading data from your graphs on the exam. Sometimes things are not in the orientation that you think they are. So speciation. Speciation is going to be kind of the creation of new species. Where do we get new species at? Okay, so this kind of brings us back to the biological species concept, which we have to know for the exam. Like, make sure you do know this one. Okay, so two organisms are going to be of the same species if they can interbreed. So that means that they're able to try to reproduce together. Okay, and when they reproduce, they have fertile, viable offspring. So their offspring are also able to reproduce, and they are healthy enough to survive. Okay, and so again, they have to be able to reproduce, and then those offspring have to be fertile and viable. Okay, and so that brings us into these different reproductive barriers or isolation mechanisms. Okay, so pre-zygotic, use the words we know. Pre is before, and zygotic makes you think of a zygote. So these are things that are going to happen before the zygote is created. So before the sperm and egg fuse together to make the zygote, this is how we're going to, of course, inhibit or stop this. Okay, so first thing is behavioral isolation. This has to do with two organisms are going to have different mating rituals. So maybe it's two birds and they have a different mating song. Maybe it's our blue-footed booby and he's got to go out with his little blue feet. Please tell me you've seen little blue feet. Oh my gosh, I love them. They're so cute. Anyway, so blue-footed booby is going to go out and it's going to show its feet off. And that's like a mating dance to kind of attract the future mate. Okay, think about penguins and the rocks. Like, so there's these rituals that they do. So if they have two different rituals, you're not going to mate with a different species because it's not doing your ritual. Okay. Temporal isolation, two organisms, they're going to mate at different times because of the fact that, well, whatever reason why. And so they could mate at different times of the day. Maybe one's in like kind of during the day and one of them is at night. Maybe different times of the month. So like earlier in the month, later time in the month. Maybe it's different months of the year. So one's in the spring and one's in the winter. Like maybe there's different times. And so because they mate at different times, they don't mate with each other. Geographic, there's actually a barrier, and because of this barrier, they cannot mate with each other. So there's something that kind of actually inhibits them from coming in contact with each other. So habitat or ecological, so maybe there's no actual barrier, but they live in different habitats. So like think about like a snake that lives in the water of a lake or something, and a snake that lives in a tree. So because of the fact that one of them's in a tree and one of them's down in the lake, they don't ever interact with each other. Like there's no barrier. The one in the tree could fall out and into the water, and the one in the water could actually come out of the water and come up the tree. There's nothing that stops them from going from each other. But because they live in those different habitats, they usually don't come in contact to mate together. Okay. Mechanical isolation, best way to say this is they're incompatible anatomically. Like they just can't mate with each other, okay? And then gametic. So usually we see this with like our external fertilization, so like a sea urchin. And so it's going to lay its eggs all on the bottom of the water, and then the sperm is going to get released and it's going to go to try to fertilize those eggs. And so there's different glycoproteins on the membrane of these eggs. And so since they have different proteins, we're going to find that the sperm can't fuse and thus it won't be able to fertilize that egg. We also have postzygotic. Post meaning after. So postzygotic means that this is going to take place after reproduction, so after the zygote is formed. So remember, the two parts is interbreed. So our prezygotic inhibits this interbreeding. It stops them from mating with each other. While our postzygotic is going to inhibit this fertile viable part of it. Okay. So first thing is reduced hybrid viability. The hybrid's just not viable. It's not healthy. And so because it's not viable, because it's not healthy, it's not going to survive. It's going to be very weak and feeble, and it's probably not going to be able to reproduce. So we won't see a continuation of that. Okay. Reduced hybrid fertility. The hybrid's not fertile. Think about a horse and a donkey. When they mate, they make a mule. The mule is fertile. Like, I'm sorry, it's sterile. Like, it can't mate. I also call them a horse, but that's okay. Anyways, so the mule is sterile. Like it's got actually a different number of chromosomes. So I can't remember off the top of my head, but it's like it has an odd number. And because it has an odd number of chromosomes, you're not going to see that it won't be able to have the homologous pairs for the for meiosis to take place. So they actually can't make their gametes. And then we have hybrid breakdown. So the first generation of hybrid is fine. They're good. They're viable. They're fertile. They're good. But as we see multiple generations, we do see that hybrid breaks down. And so the whole point of this is to keep the species different. Okay. So the two species are not going to mate with each other. Okay. Yes, I do know that we sometimes get species. We somehow get new ones, but that's okay. So sympatric speciation is how we can create new species in the same area. Okay, so new species of the surviving ancestral species, they both have the same geographic area. So this is usually due to some type of absence of gene flow. Okay, so the big example we always use is about these flies. Okay, so the hawthorn flies, they were on the apple tree versus the berry flies were on another tree. And so because when the new one was introduced, half the flies went to a new habitat, they stopped mating with each other. So usually this is our habitat isolation. Is also could do behavioral isolation. Maybe there's some reason why some of them started doing a different behavior, so they stopped mating with each other. Sexual selection, so now there's some trait that's more favorable, and so some of them will pick that trait. And then polyploidy. In case you don't know what polyploidy is, this has to do with that there's more than two sets of chromosomes. So maybe there was some error that took place during meiosis, and that error led to this offspring being, I don't know, a tetraploid. So the tetraploid individual is not going to be able to mate with the diploid individual, and so they become their own new species because these two can't mate with each other anymore. Allopatric has to do with it. There is a geographical barrier. There's some barrier, and because of that barrier, we have speciation taking place. So this happened back in the day. Colorado River separated. I think they were squirrels. They were the check monster squirrels. Anyways, but so the Colorado River separated the two. And so because of that, they couldn't mate with each other because the squirrel or chipmunk or whatever it was couldn't get across the water. And so there was an absence of gene flow. And so now that the Colorado River dried up, and we've, of course, Grand Canyon is there, and they can come in contact with each other, they're not able to mate because now they're actually two different species. Okay, and so here's that. I think it's a squirrel. I don't really know. It's okay. So that was all the content from Unit 7. Like that was, of course, quick review of it, only 30 minutes, but you got there. Okay, so let's do some multiple choice practice and some free response practice. So by discharging electric sparks into a laboratory chamber atmosphere that consists of water vapor, hydrogen gas, methane, and ammonia, Stanley Miller obtained data that showed that the number of organic molecules, including many amino acids, could be synthesized. Miller was attempting to model early Earth conditions as understood by 1950s. The results of Miller's experiments best support which of the following hypotheses. Okay, so we're trying to say, okay, he set up this experiment. He had electric charges that came in. There was originally water vapor, hydrogen gas, methane, and ammonia in there. And he was able to somehow, without life, make organic materials. Okay, so we want to read through these to see which one of these sounds like something that could have happened in there. So molecules of essential life today did not exist at the time Earth was first formed. Okay, that is a true statement, but it's not something that we got from the results of his experiment. Okay, the molecules of essential life today could not have been carried to primordial Earth by a comet or meteorite. Well, he's proving that we're able to make these things in the old Earth. So I wouldn't go with B either. C, molecules of essential life today could have formed under early Earth conditions. He proved this, right? We had our amino acids and the things that are part of life today had been synthesized. He used early Earth's environment where we had the water vapors and the electric charges and all this, okay? So I think C is good, but let's keep rolling. The molecules of essential life were initially self-replicating proteins that were synthesized approximately 4 billion years ago. So again, although that is a true statement, it's not based on the data we have here. They're wanting you to look at the results from his experiment. Okay, so making sure that you are using the information given. So of course, that is off. It should be C. I promise it's C. So apple maggot fly is native to the North American and initially fed on fruit of the wild hawthorn. Since the mid-1800s, a population of flies emerged that instead of feeding on domesticated apples, apple maggot fly typically mate on or near the fruit of their host plants. Many varieties of apples ripen 3 to 4 weeks before the hawthorn plants. Okay, so the food that they're eating are going to kind of ripen at different times. So they're going to be eating and on those trees at different times. The different fruit preferences of the two fly populations will most likely have which of the following effects. The flies that eat hawthorn fruit will increase in number, while the flies that eat apples will decrease in number because of insecticides. Did we talk about insecticides? No. The single fly species will evolve into 2 distinct species because of the lack of gene flow between the two populations. So if this group is going to that plant to eat and this group is going to that plant to eat, are they going to be in the same place together? Well, no. And so I do see a lack in gene flow. Let's say I like B so far. C, the ability to survive on a diet of 2 different fruits will help the flies to learn to eat many more types of fruits. That does not have anything to do with what I'm talking about. The flies that eat hawthorn fruit will lay some of their eggs on the earlier ripening apples to minimize competition among larvae. Although that sounds like a true statement, that really doesn't give us the effects that we're seeing. So of course, our answer is B. Hey, and my thing had it right. The reason why there's errors is because I pulled it from one PowerPoint to the other, and I changed the font to make it cuter, and that's, sorry, y'all. So here we have the data that we have for the presence of different traits. So if it has a presence, it's a positive. If it has an absence, it's a negative. Of these different traits in the different species are shown in this table. We need to figure out which of these is a representation of the data. And this is how you might see these phylogenetic trees or cladograms on the exam, is they may have you have a character table and you've got to pick out the right one. Or they may give you a blank cladogram that you need to fill in yourself. Okay, so looking at this data, I can see that one of them kind of stands out. Y stands out. I see all of these negatives. So Y is missing all the traits. And so I would call this my outgroup. It doesn't share any of the characteristics with the other individuals. Okay, so that's going to show that Y has to kind of come off on itself. So Y is right here by itself, Y is right here by itself, and then Y is kind of right here by itself. So that's going to help me to eliminate B because of the fact that in this one, I see that Y should be closely related to X, and that's not what I see based on my data. Okay, so that one's gone. And then, of course, I see this one gone because it's not closely related to the V and the X. Okay, so now if I look here at X and Z, I can see that X and Z are closely related. They both share character 1, but they don't share character 5. So I would find them kind of branched together. If I look here at C, I don't see that the X and the Z are sharing a branch together, but over here I do see them, okay? And so that shows me that A is my answer because of course we have these two that are closely related using that X and that Y. And then you also see that the V and the W are closely because they share 1, 2, and then 3 is where they differ. And then of course they share the same thing for 4 and 5. And so I know that that is my favorable one, or the one that's the right one. So amino acid sequence of cytochrome C was derived for 5 different species of vertebrates. The table below shows the number of species, differences in our sequences. So using the data in the table, we want to create a phylogenetic tree on the template provided to reflect the evolutionary relationships of the organisms and then provide reasoning for the placement on the tree. Okay, so if they give you a cladogram on your FRQ, it's going to look like this. They're going to give you a blank one that you have to fill in the information, and of course they'll give you data. Okay. There was a phylogenetic tree on last year's exam where they were modifying it. Okay. So I do not predict that you will be doing one this year, but there could do the same thing 2 years in a row. Sorry. But so what's going to happen is that if this happens, then question 5 is where you'll see you having to modify. That's where they always have you modifying a diagram. And so make sure that you fill in the one that is in your book. Okay, so you're going to have a question book and you'll have a response book. So make sure you're doing your graph for number 2 and your whatever modification you're doing on question 5 in the book. Don't do it in the questions because your readers will not see the questions. Somebody just said, when am I doing my Unit 8 review? It's, I think, next weekend. I think I'm doing like literally every weekend until the AP exam because that was the only way I could fit the schedule. All right, so how can we fill this in? So I said before we wanted to look at the ones that were most closely related. So if I look here, I can see that there's a number 1, okay? That would be between my E. ferris and my E. africanus. So I'm going to put those on the branch right here. They're closest together. That branch kind of is nice and tight, so we know those are there, okay? Now, is there another number that is small in this chart? Well, I see right here, there's a 3. G. gallus and A. forestry, which, P.S., that's a penguin, y'all. Isn't that so cool? They put a penguin on the exam. I know it's just for me. It's okay. Anyway, so we know the G. gallus and the A. forestry, or whatever, however we say that, are closely related, so we're going to put those together. And then the one that is most similar from them all would be D. polyptus. So if you look at D. polyptus, it's 18, 17, 20, and there's another one, and 21 away. So you can see that one has the most differences. So I placed it here because it's the out group, because it has the most differences in its amino acid structure, okay? So part B says identify whether morphological data or amino acid sequence data are more likely to accurately represent the true evolutionary relationships among the species and provide reasoning for your answer. So as we said earlier, the one that is most favorable and the one that's going to give us the most accurate is of course going to be amino acids or our molecular data. Okay, because of the fact that morphological data could show convergent evolution, could show things that are similar due to a similar selective advantage, not because they have common descent. Also, we're going to see direct changes that are taking place. A mutation, they had the same DNA, a mutation took place, and now there's 1 or 2 differences from them. So they share that genetic code. Okay, but College Board was really nice on this one. And so they said to identify whether one or the other. So you have a 50-50 shot here, right? So on the exam, if you don't know something and they're giving you 2 options, just guess. If they say predict what's going to happen to something, increase, decrease, stay the same. Like, make a prediction. Even if you don't know, try. You could accidentally get a point. But College Board, as I said, was really nice on this one. And they actually accepted either one of them. You could have also said morphological data. So on this one, no matter if you wrote morphological data or amino acid sequence, you still got 1 point here. So that's awesome. Okay. And so the reasoning they said sometimes our molecular sequences could result in different morphologies. So even though they may have a similar DNA structure, they could still look different. And then an example of the fact that you have similar proteins but different morphology. So thinking about like a chimp versus a human, kind of looking at that morphological data. Okay. So here we've got these guppies. So we have these adult male guppies. They exhibit genetically determined spots. So the spots are due to genetics. In a study of selection of males and female guppies from a genetically diverse population, they were collected from different streams and placed together with no predators. So they're all together. Okay. Study maintained for several generations, isolated area before separating them into 2 groups. You can see here we've separated them into the groups. One group is going to be where there's a predator. Another group is going to be where there is no predator. And I think I just labeled the wrong ones. That's okay. At different times, we then determine the number of means. The vertical bars represent the errors of the mean. Okay. So thinking to yourself and thinking about your teacher and how much they have honed into you, they have always talked to you about these error bars. And they said, if the error bars overlap, that means that the data is not significant, right? And so what we're looking for, of course, is error bars overlapping, okay? But what does the error bar actually tell us? The error bar is showing us the difference in the data, like so the stretch or the range that the data is, okay? So right here, when we're at 0, they're a genetically diverse population. There's a lot of differences in the DNA. So we're going to see a lot of differences in those, the mean number of spots. There's a large variety in them, okay? Versus if I have a smaller SEM bar, that means the data is very kind of concisely together. Okay, so there's less differences in my data. So that means that most of the fish are going to be kind of in that 12-ish area. Okay, so on this question, we're not looking for the error bars to overlap because what they're going to ask us is describe the change in genetic variation in the population between 0 and 6 months. So between 0 to 6 months, I see that at 0, we had 10 spots. At 6 months, I have 12 spots. So I'm seeing that my variation in my data is that, oh, we got more spots. But that's not what they're asking you. They're asking you about the actual DNA. They're asking you about the stretch of that bar. And so I see that my error bar goes from being very large to being very small. And so because of that, my genetic variation is actually decreasing. Okay, so my genetic variation decreases because my SEM bars got smaller. So be very careful not to fall into traps. Okay, they're asking about the range of the SEM bar. They were not asking about the number of spots. Okay, so be very, very careful there. Part B, propose one type of mating behavior that could result in observed change in the number of spots in the guppies between 6 and 12 months in the absence of a predator. Why is that in the wrong spot? I can't do that. I'm sorry. Okay, there's fix. So in the absence of the predator, we see that it is increasing, right? And so when the predator is not there, okay, why do I see an increase? Well, it has to do with the fact that without a predator, we're seeing sexual selection, okay? The ones with more spots are going to be selected for. We're going to see more of those females selecting for those. And so we're going to see the, of course, increase of those number of spots, okay? Versus random mating behavior, we'll see just maybe randomly they mated and that caused there to be the increase, okay? Part C, evolutionary mechanism that changes over time. Oh, good, I have the right one. 6 to 20 months in the presence of a predator. Wait, I said that backwards. Anyways, no, presence. Okay, so here we see it's decreasing. And so why would I have these spots decreasing? It has to do with the fact that since the predator was there, spots are going to allow them to be seen. And because they can be seen, we're going to see that they're going to, of course, consume them. And so directional selection against the individuals with large spots, directional selection for individuals with fewer spots. Natural selection will be used as long as you use it in context, talking about that the predators could see them and would consume them. And so they're less likely to survive and less likely to pass on that trait. And so we see, of course, the ones that have less spots are more likely to survive and more likely to pass on those traits. We see the decrease taking place there. Whew. So what questions do you have? And so while you're coming up with your questions and throwing those into the chat, I'm going to go ahead and tell you about a couple of resources I think would be helpful for you. So check out Marco Learning. Marco Learning has tons of YouTube videos, not just for bio, but for other subjects. And so go check them out. They're the couple weekends before the AP exam. We're going to be doing some live streams on there to help you review for the AP exam. And so check out Marco Learning. Absolute Recap. Okay, Melanie Kingett from the Absolute Recap. She has a podcast that you can use. So if you like to go for a walk and listen to your content, go check her out. She has, I think, government, physics, chemistry, biology, and music theory. So check her out. And then Edge.com is a new platform that's kind of getting started. And so what's really cool about them is that they're going to kind of email you each day. And so like if you were to do these topics, right? So if let's say that's our answer and you're not really sure about it, it's going to tell you kind of additional information to kind of back it up. And then whether you mark it correct or incorrect is then going to determine if it's going to cycle around. If you get it correct, it's going to cycle it a couple days versus if you get it incorrect, it's going to cycle it more frequently. And so this is kind of using the same kind of technology as Duolingo and is going to try to kind of help you to better understand. So this is kind of a good resource that you can just use each day to do some quick review. And as I said, they do have like some information on there that can kind of help you to better understand. Like they've given you some pictures and it's based off of the OpenStax textbook. So if you're needing kind of like a textbook, they work really closely with the OpenStax book. Okay. And of course, don't forget, I've got review on my Instagram. I post TikTok. Well, TikTok's still around, although I did hear that they just passed the law where they're not going to have it anymore. And of course, you're already on my YouTube, so we're good. Okay. So what questions do we have? I see, when will I do my Unit A review? That's going to be next weekend. I do it. I believe it's 3 o'clock, same time. Can I explain how error bars represent DNA variation? Yeah. So with this, what we were seeing was the variation in the data, the DNA, okay? So this is where the mean population is, right? So if we were to take every single like piece of DNA, right? And we were thinking about, okay, this individual has 5 spots, this individual has 4 spots, and how many spots do they have? And we averaged them together. The average was 10. But there was a large stretch of the data. And so I had a lot of them that were in the 2 range and a lot of them that were in the 10 range, which I probably should use the example, but a lot of them were in the 8 range, which a lot of them were in the 12 range. And so since there was a large spread of my data, that gives me a large genetic variation, right? There's a lot of differences here. versus over here, they were all in that 12 range. And so that shows me that there's less variation in that DNA and less variation in the DNA. Okay. Do I have any tips on how you should be studying? So at this point, we have 20 days till the exam, and I'm not sure when you're watching this video. If you're live, of course, it's 20 days. If it's later on, then you have however many days. There's always a countdown on my website. So you need to be kind of more spot checking. Okay, so what is it that you struggle with? What topics are hard for you? And if you're needing like more of a holistic, like I don't even know, I'm just gonna look at everything, then watch somebody's YouTube video. So either watch one of mine, you could watch Boson Bio Crash Course, you could watch Rob Tate, you could watch my kid's favorite Poser. What's his first name? Gabe Poser. So there's a ton of YouTube people out there. There's this Mike Chipman. There's a lot of different YouTube teachers. Okay, you can use any of them that you want. Okay, watch a unit video and kind of get a quick review. You also probably want to be looking at multiple choice and free response. Now, can you do every single free response between now and the exam? I don't know how many AP exams you're taking. But if you can, I would do at least 1 or 2 FRQs a day just to kind of help you get that practice in and get you ready for the exam. If you're needing a spot check on something, go find, again, a video on that, or go look up, go watch the AP Classroom on that. See if your teacher will open up the topic questions for you or the PPCs or the performance progress checks. Get them to open those up for you. If they open those for you, then you can, of course, practice those different topics to review. And those are the same people that are writing the AP exam that are writing the PPCs. So that's the best multiple choice practice you can use. And then if you can convince your teacher to proctor a mock exam, that's going to be phenomenal for you. So 1, overview different YouTube videos. 2, work on multiple choice and free response practice. 3, take a mock, and 4, do some like specific studying on your weakest subjects. So somebody asked where that is. I think I actually like completely closed it. It was etch.com or etchlearn.com. And so that's where that was. I think I had logged in before, so you could have seen it like full. But they also have like an AI that they're working on. And so you can like ask it questions, but sadly that costs money. Like you could like, buy it. I think it's like $9 for a month or something. So it's not like too crazy. But they do have that if you're needing it. Yeah, anyways. So what is the difference between bottleneck and genetic drift? Bottleneck effect is going to be where we have a large population or a population that gets drastically decreased by some type of natural disaster. That is a type of genetic drift. Genetic drift has bottleneck as well as the founder effect. Okay, and so bottleneck was an example of it. And so since I had that drastic decrease, my new population is not representative of the original population. And I think that is all the questions that we have. No one else has posted questions in there. So sorry for all my issues today with my errors on my PowerPoint. Remember, AP Bio Pain was just success, and I hope y'all